I think today we're going to switch to have the committee updates following the presentation, because we have our presenter calling in all the way from Sydney, Australia, I believe. So it's rather late there this evening. So we'll probably start with her presentation so that she can get to bed before midnight, hopefully. So I believe that Anne, you are going to introduce our speaker for today.
Yeah, thank you so much, Rob, and thanks, Sophie, for joining us so late in your evening. We really appreciate it. So just a little bit about the presentation today. The presentation will be by Bioplatforms Australia. Bioplatforms enables Australian life science research by investing in state-of-the-art infrastructure and associated expertise in the specialist fields of genomics, proteomics, metabolomics, and bioinformatics. Bioplatforms catalyses research collaborations to build new capabilities and critical data resources supporting some of Australia's biggest science challenges. These challenges span agriculture, biomedicine, and the environment, as well as extending to relevant international endeavours. Since the program's inception more than a decade ago, continual refinements are being made to the data management strategy to ensure the omics resource meets international best practice and the FAIR principles. Sophie will present an overview of Bioplatforms' data strategy and future outlooks. Sophie has 18 years of research experience working across diverse environments in Europe and Australia, including private institutes, university and industry-oriented research groups. She has expertise in molecular science, biotechnology, and microbial genomics, and has participated in several large European research consortia. As a program manager with Bioplatforms Australia, Sophie facilitates scientific collaboration through their national scientific initiatives, providing a critical link and greater engagement between their national infrastructure network and the initiative consortium members. So with that, I'll pass it over to you, Sophie, and thank you so much for being with us.
No problem. That sounds very grand for a glorified cat herder. I am sometimes calling myself. But welcome, everyone. Hello. Yeah, it's a little bit late, but it's not too bad. I'm usually a night person, so that's all right. I will try to share my screen. Just hopefully everything will work properly. Let me try this. And I am, can you see my slides?
Not yet, no.
That might take a little while to come up. Oh, okay, no, let me just swap a few things, share screen. That will be better if I do this like that. Let me do this one, share. Now that should be a little bit better, and I will put it. Can you see it now?
Perfect, thanks.
Yep.
I'll try to put it on the main one. Do you see my actual slide and not the presenter slide? It's always the thing with the Macs because I'm on.
Yep, you're good.
Perfect. Let's start then. So yes, I am a project manager. I'm working with Bioplatforms Australia, and I thought I would do a little introduction on what Bioplatform Australia is, despite the fact there was a little introduction in the abstract of my talk, because it is something that is quite different to a lot of places internationally. So Bioplatform Australia is a not-for-profit organization. We were established in, let's see if I can change that. Okay. We were established in 2007 through funding from the Commonwealth, so the federal government of Australia, and through a scheme that is called the National Collaborative Research Infrastructure Strategy. Hence, we just shorten it by NCRIS because it's a little bit of a mouthful. But what it is really fund that the federal government is giving several entities and our sister entities to provide infrastructure for all the research community. And that infrastructure is really accessible not fully freely, but it's made to provide an infrastructure that is really shared and national. And really the collaborative is a very important word. It is not a competitive process for this access. So our team is obviously a small team. So our core team is 6 people. We have facilities all around Australia and also a lot of staff in those facilities with a quite substantial investment of capital. And that capital is for both the capital of getting that infrastructure and those shiny machines, as well as the operational support, so the personnel and the expertise of those people to use those machines, and in all the omics environments, so genomics, proteomics, any omics that you can think of, as well as synthetic biology. We are now going into that infrastructure, and obviously, anytime you speak about omics, you want to have some bioinformatics. So we are also supporting the national infrastructure on bioinformatics for Australia. And when I mean national, it's just putting arrows to make it a little bit bigger, but we are actually trying. Australia is big. If you take that, it's 10 times France, it's actually very large, but we don't have that many people. It is really concentrated around the coast. So we try to have facility in all the major capitals. And it is the facilities in both the genomics and all the omics. So we do have them a bit everywhere. We have now started that the synthetic biology infrastructure. That's why there's only 2 of those. And they are a bit concentrated, but it's sort of a new step change from our activities. The one thing we actually found very early on is that infrastructure, mostly in the omics area, it's not only the shiny machines, it's actually also the data and the resource. And what we did is actually to create those framework initiatives, which were the deployment of all the platforms we were supporting, and creating those framework initiatives as resources that were going to be open access and a collaborative work through the whole research community. So none of these are done in the traditional way, just having to present a grant and having to write a full applications and then getting money given to you to do something. We actually create these as a full inclusive collaborative aspect of work with people from all over the country. So it is obviously very targeted to Australian needs and Australian challenges. But so far we have 3 themes of work which cover most of the biology type work. It is in the health, the agri-food and environment. So each of those we sort of tend to have them quite divided. Obviously, sometimes they get a little bit of crossover, in particular when you start talking about pathogens and so on. So we have done that for now more than a decade. Obviously, that was a bit of a steep learning curve, but we have completed quite a lot, and we established those initiatives looking at really both a national challenge needs. So we wait for signals from all range of stakeholders, and we make sure to have a complete inclusivity of who participates to this. There is always a governance and a structure, but we try to make sure it is as open as possible because we are federally funded, so it's public funds. And we also ensure that there is co-investment. So we obviously put some funds towards that initiative and creating that data resource. But we also get funds or co-investment either in cash or in kind. And usually a lot of researcher and organization put a lot of in-kind data to gather sample and so on, as I will talk a little bit later on. So really, those initiatives, they have really key outputs of gathering really broad community. So it's not like, if you have a research grant, a lot of time, it will be a small group or a team, sometimes much bigger. If you look at Horizon type grants, it's actually much bigger, but for our one, it is always a cross-section of a whole range of organization. We never create an initiative that will be from one team or from just one organization. It is always an initiative that will have 40, 50 organizations and sometimes well over 200 participants. So it can need a little bit of structure to actually run. And we create that National Data Initiative, and this will look at not only the reference omic resource, but also the way of creating and generating that resource, the protocols, the guidelines, ensuring everything is best practice and etc. As well as sometimes creating also the collaborative software and tools that are needed to analyze that data or just go further with that data than just generating the data and making it accessible. I will touch on that in one of the initiative that we have, which is really interesting. You might have heard of as well, the Threatened Species Initiative. So those collaborative software and tools are usually gathered in part of the bioinformatic infrastructure that we support. Australian Biocommons is the lead organization for that infrastructure, bioinformatic infrastructure. And we also have a Galaxy instance that we've actually evolved quite a lot and have some nice links with the data and so tool available. And all of these really is giving a nice platform, a nice stepping stone for all the research community, all the organization to use and to analyze and actually have really efficient application of the data, not only just creating a data resource for the sake of creating a data resource, but to actually use it for something like a threatened species management, like an industry application, and so on. So I will talk mostly about the biodiversity part of some of our initiative, because sort of I wouldn't say the more developed, but the one that is sort of easier to show in details. So we have so far done quite a lot of them. So we started through some of the soil work, actually, because it was sort of a little bit more, it's always been there. The genomics there was actually a bit simpler, if someone can say so. And then we have gone into creating pretty much initiatives for nearly every single large taxon of vertebrates. And we are now having, and plants, obviously, and we are now sort of attacking the insect and the invertebrate space, which is quite complicated in Australia because there is such a wide variety of invertebrates that it is much broader and much more complicated to create that resource. And really, as I was saying before, what we do is really working on the best practice and best standards to create protocol and the data resource and sort of sharing that data and making sure that it is going to be possible to be translated and encouraged to be translated. So this is a little bit, so this is my little garden lizard, is in my home. And this is a stone water dragon. It's called Lizzie, despite the fact he's a boy, but that's okay. The referential genomic information is all on pretty much all the taxon groups. We have done some invertebrates already, crustacean, mostly threatened species one. And we generate reference genome. We've already got about 120 planned or on the way. We're adding more every time. And we will certainly, towards the end of the year, be close to, I would say, 170, because we're adding some more now on threatened species in particular, and for the birds and fish. We're not only doing the reference genome themselves, we're also adding phylogenomics data sets to just try to get the taxonomy correct and the phylogeny correct. It is usually done very well in the Northern Hemisphere. Australia is sometimes a little bit different because it is so broad that sometimes we just discover a whole range of species and taxon complex and species complex around that nobody has been trotting across. So it is still something very strongly done. And we do that also with an international partner. For example, the plants. There was a very strong work with the PAFTOL group, with Kew Gardens that worked with recreating the whole phylogeny for angiosperms, flowering plants. And they've now, with collaboration, succeeded to do up to 93+% of that whole phylogeny. So it is a very nice work. And lastly, but not the least, we also do some reference genomic resource for conservation focused aspects. And this can be resequencing, but very often it is based on SNPs or genotyping. And we are doing this through right sequencing, so the sort of restriction digests, and establishing SNP arrays. It is partially to make it more amenable for organizations who may not have the expertise of dealing with resequencing of thousands or hundreds of organisms. So it is still very important to have those genotyping type activities. And all of these resources that we're creating, I was mentioning that they were really trying to be done in an aspect of having a use, not just creating that resource. And a few years ago, we worked with the University of Sydney, Caroline Hogg, who we created that Threatened Species Initiative. And this is really not the primary purpose of creating a resource, but creating a resource to assist directly some conservation efforts and some activity. So none of the project or the reference genome or the genotyping, the population genomics we're creating here is going to be created just to be stored and made available. It is always done with a conservation partner, and they've already had some great success of creating those populations to ensure translocation, selecting the best animals, or breeding program, and so on. So this one is not a taxon specific, it is a threatened species specific type of species, and that's where we had invertebrates and vertebrates and plants. So it is quite a broad initiative. And for this one as well, to assist with the actual outcome, there is a data portal, more of an analytical data portal that is being created with that resource that will then be available for the conservation partners to use. So it is generated as a sort of a service, for the moment a free service, where they can put their information. So for example, if they have a couple of birds, they want to go, they do an analytics or genotyping, they put all their raw data, they don't really know how to analyze it, but it will just go through a genomic pipeline that will provide them with a species management report that is nicely done through their needs, and that's been realized with a lot of discussion with them and with what management partners need and so on. So it is really not only the genomic resource, but pipeline and tools that are going to assist on the longer term to have real impact in using that genomic data, which would not have been possible without that genomic data. So that is when I'm done, but to create a genomic data or any omic data, and that is usable and can be trusted and can be reused in the long term, obviously there is the main part, which is usually sometimes the most complicated part, is all the metadata. So, it's good and well to create terabytes of data if you don't know where it comes from, how it's been created, how it's been analyzed. It's actually pretty much useless, and it will just stay and not be used or being used for one type. So what we do for every single one of our initiative and every single one of the data sets we produce, we have a very extensive range of metadata, and the metadata stems from, goes from the actual sample origin, how it was taken, where, what it is, any detail for the soil and microbiome type work. We actually record the environment details in chemical, physical terms. For the organism, it's sort of a little bit more the organism itself and the specimen. And it is usually about, for the sample, we have about 70 fields. For the library, or what we call, which is the processing part, we have close to 15 fields or more. And then we also details how we manage and record the data. So, when it was taken in and so on. So obviously, this is good and well. We also need to make sure that we have, and we're working on that very much more, the recognition of attribution and rights, not only from the people who are working on it, but permits if it is taken from national parks or restricted areas. Obviously, the custodians of lands, there is all the indigenous recognition of information. And all this is curated, managed through our data portal, which I will talk about. And we also obviously try to follow international best practice and standards. So that means that the curation is constant, because unfortunately or fortunately, the standards and practices change, get better, get improved, and all of the data needs to also be improved to that standard to ensure that we go through the FAIR principle of findability, usability, reusability, etc. And also, if sometime we need to add data just that was missing. All of this is put in our beautiful portal, which is managed by one of our team in Queensland, the Queensland Cyber Infrastructure Facility. And it is really a record. It's a data dump, you can say it. It's a pretty data dump, but it is getting all of the data organized, managed, linked, everything with all that metadata, and then placed as open access. We do have an embargo, small embargo period, just because a whole lot of the researchers are putting a lot of effort and a lot of co-investment. So we give them usually a year of embargo, and then the data is becoming then open access, with some restriction, obviously, for sensitive information, like threatened species, we don't give any coordinates and so on. All of the data, the records are in standardized file name structure. So again, it sort of increases the reusability of the data. And as I mentioned, the best practice metadata to ensure that. So that was just a little bit of that. It is all based on SICAN, so which is very simple data storage, and also sort of data infrastructure presentation. We use Amazon S3. So we always like to say it's very simple. We take all the information for sample processing, put that in some beautiful tracking sheets, ingest it, and it's all pretty, and we get all the information. my devs on the team of Data Portal tell me it's a little bit more complicated, but I'm just not looking at the details. As long as it works, it's fine. We do work a lot with the Amazon S3 and all those report, and so we just keep that in there. So I won't go too much in detail in that one. I don't understand all of those. I just manage the team that just does that. And the one thing we also started, so we started for a long time to get that data portal and work on it and make sure that it was fit for purpose to the best of our ability. One thing that we have started for the last few years is to ensure that the data was also linked to other capability. That is either analytics or indexing of all sorts. So, really being able to be either harvested or used in other platforms. And the Australian Biocommons, which is the Australian Life Science Research or Infrastructure for Bioinformatics, is providing quite a lot of those tools and softwares, and we're making sure that all the data we have in the data portal has got direct links. So, people don't need to always just download the data, which is sometimes terabytes, and then re-upload it somewhere. We're actually creating those nice direct links behind. We have a whole range of analytics services and annotation services that are provided mostly freely to the Australian research community, which is usually not the case if you don't pass through the NCRIS collaborative strategy or scheme. And there is a whole range of other type of support and training available through Biocommons. And also in terms of, so that was the analytics, we're now doing something which is sounding a little bit like ERGA, but it's ERGA, and just to make the variation. So it's the Australian Reference Genome Atlas, which is also, again, a collaboration from several organizations. Most of those are actually those NCRIS funded sister organization or funded by us. And this is really an indexing service that we're just starting to work on. Very often, I know that a lot of time GenBank and SRA and all of those places do have all the data sets, but for Australia, maybe because we are far away, we are big, some of it goes into those big international, some of the data we have goes into international repositories. Sometimes it is hidden into some very strong places, and it's actually really hard to gather really Australian specific data. So we have started to work on this one to really make that indexing service, which is targeted to Australian sourced data sets, and they obviously look at GenBanks and so on. They look at our data portal, and they also, through there, we're just trying to encourage people to also deposit the data that sometimes and very often in Australia is being, gathering dust in some hard drives behind in the offices of some professors. So this one is actually very much underway. It's going to be launched very soon, and we can just select all that. We have obviously all the metadata available. You can select and sort of filter all that data. So it's actually a nice indexing service, and also there is a way of just directly going and sort of helping with gathering the data itself and sending it to analytical services as well. So which is really going to help a lot the research community, and sometimes research community may not be as versed in going in NCBI and gathering that data themselves. And finally, So I'm going a bit over now, talking too much. We are also working, as mentioned, working with international omics efforts, both in our initiative like PAFTOL and NMDC for our microbes. We are also working or aligning our activities with the Biogenome Project, GOAT as well. We might have somebody on the board of or advisor or something, and Elixir through Biocommons. So we're very happy to just both, share knowledge as well as obviously, benefit from it. And this is me.